26th of August 1976, in a village named Yambuku in the Mongla province of the northern part of what is today the Democratic Republic of Congo, and at the time Zaire, a man named Mabalo Lokela, the headmaster of a local school, arrived at the local Yambuku Mission Hospital with symptoms of an unknown disease. The care available at the hospital was basic but an important lifeline in the region, serving Yambuku and several other surrounding villages and offering 120 beds. As a volunteer mission hospital run by the Catholic Church, the staff consisted of 4 Belgian nuns, a priest, 1 Zaire nurse, and 7 Zairian men. Mabalo had recently been on a Christian mission near the border of the neighbouring Central African Republic and along the region of the nearby Ebola River. Judging from his symptoms of chills and a fever, the staff began to suspect that he had contracted malaria, a common problem in the region, and gave him an injection of chloroquine and an antipyrectic. A few days later, on the 1st of September, Mabalo returned to the mission hospital in significantly worse condition, now suffering from a high fever. Advised to rest, he returned to his village Yalikonde, only a kilometre away. By the 5th of September, he was in a critical condition, now bleeding from all orifices, vomiting, and suffering acute diarrhoea, as well as displaying symptoms of chest pains, headache, fever, agitation, and showing signs of confusion. Whatever he had, it was clearly worse than malaria. On the 6th of September, he unfortunately died as a result of his symptoms. From the time of Mabalo's first admission to the hospital to the weeks after his death, several other patients began to display similar symptoms. Cases of what were first thought to be malaria, typhoid, or yellow fever soon began to follow his same brutal trajectory. Many of the patients had either previously received injections at the hospital for other diseases, or had had close contact with a previous case. Even at Mabalo's funeral, of the family and friends that had gathered to prepare his body by the traditional methods for burial, 21 would later fall ill and 18 died. With a terrifying speed and lethality, this unknown disease began to tear through the local community, seemingly able to jump from patient to patient with ease, centred around the mission hospital. By as soon as the 12th of September, the situation was clearly escalating. With no doctors or equipment to make a diagnosis, the hospital and community made an urgent plea to the Central Zaire government for assistance. Dr. Ngoy Mashola, the chief medical officer of the Bumba zone, arrived in the village on the 15th. Over the next 4 days, he provided the first clinical description of the new disease. In his daily log he wrote, quote, "The illness is characterised with a high temperature of about 39 degrees centigrade, hemiostasis, that's vomiting of the blood, diarrhoea with blood, resternal abdominal pain, prostration with heavy articulations, and rapid evolution of death after a mean of 3 days." He estimated that from the period of the 5th to the 22nd, there had been around 30 cases and 22 deaths in the hospital from this disease. On the 23rd, a larger national team arrived led by Dr. Jean-Jacque Mayumbe, a Belgian Zaire microbiologist who'd been working as a field epidemiologist for the last 3 years, and Colonel Amombo, an epidemiologist. By this time, the hospital was almost abandoned as patients began to avoid contact. On his arrival, Jean-Jacques recalled finding only a mother and baby in the hospital, the latter of whom died during the night. By morning, patients began to arrive for physical exams after hearing the central government had sent a medical team. Conducting these exams, Jean-Jacques noticed an unusual tendency for patients to bleed profusely after taking their blood, something he hadn't seen before. Additionally, he noted with concern that several of the Belgian nuns ill with the disease had previously been vaccinated for yellow fever and typhoid, which could either rule these diseases out as possible sources of the infection or indicate a new, more deadly strain. The race was now very much on to identify the disease, or if it couldn't be identified, at least learn more about it. Jean-Jacques began to take blood samples from the liver of patients with a long needle, noting again the unusual continuous bleeding this would cause. The real challenge would be getting a live sample to a microbiology lab well equipped enough to examine it. Although Jean-Jacques had obtained his PhD in Belgium at the Riga Institute, in 1973 he had returned to Zaire in order to better contribute to the newly independent nation's healthcare system. However, he soon found that he simply didn't have the resources he needed to properly contribute, lacking both a modern lab and mice for experimentation. It was this lack of equipment and resources that had led him to take up the role of a field epidemiologist in the first place, which simply highlighted the challenge. One day after arriving, on the 24th, he was able to persuade one of the patients, a 40-year-old Belgian nun working as a midwife in the hospital, to return with the expedition to Kinshasa via 2 commercial flights. Once there, at the university laboratories he analysed the blood samples, finding inconclusive results for yellow fever and only a partial positive for typhoid, but the results were less than clear. On the 28th, a Belgian physician, Jacques Courtil, took a series of blood samples from the same nun, who by now was near death. Jacques packed these samples in vials in a thermos flask full of ice and included a handwritten note describing the condition of the patient and noted they suspected the cause to be yellow fever. The thermos was then, quite incredibly, flown by hand luggage on a commercial flight to Belgium, where it was delivered to the Institute of Tropical Medicine in Antwerp on the 29th, arriving with several of the vials broken and infected blood flowing through the melting ice. Despite this, enough samples had survived for study. 3 scientists in the microbiology department inoculated the samples into virocells, kidney cells from an African grape monkey commonly used in research. Under observation from an electron microscope, Wim Jacob observed for the first time what has now become a world-famous image: a large, worm-like structure, gigantic actually in terms of viruses, present in the sample. Only one other virus had ever been found that looked similar: the infamous Marburg virus, a disease that had infected 31 people in Germany and Yugoslavia in 1967. This disease had been associated with lab workers working alongside infected monkeys imported from Uganda. Knowing the seriousness of this, the team alerted the WHO, the World Health Organization, who immediately requested that all samples be sent to the microbiology research establishment in Porton Down in the UK. Although the Belgian team did send some of the requested samples, arriving on the 5th of October, some of which were then in turn forwarded to the American Center for Disease Control and Prevention in Atlanta, they were also angry at having been forced to give up their work. Although they lacked the containment facilities available in the CDC, they did not want to be left out and retained several samples for their own work. Another set of samples were also sent from Zaire to the Institut Pasteur in Paris, but these were all forwarded on to the CDC. Between the Belgian, British and American labs, a raft of isolation and characterization tests were performed that demonstrably showed the new disease was something different to Marburg, closely related almost certainly, but new. 2 members of the team from the CDC, Aline Harrison and Fred Murphy, took the now iconic electron microscope photos of the new virus that published alongside their research. The new name for the disease, Ebolavirus, was taken from the river where it is believed that Mabalo contracted the virus. Okay, so it's here that we're going to have to take a step back from the human story behind Ebola in order to talk about the disease in a bit more detail. First, I want to give you a very quick overview of the virus itself. Ebola is the common term used in media to describe the actual disease caused by Ebolavirus. It can also be known as Ebolavirus disease, EVD, or Ebolavirus hemorrhagic fever, EHF. Viral hemorrhagic fevers are essentially diseases that target the organs or the cardiovascular system. There are quite a few types, including yellow fever, Marburg and Ebola, and their disease is generally caused by about 5 distinct families of RNA viruses. One of these families, filoviruses, is the one that Ebola belongs to, along with some quite closely related diseases such as Marburg. Today, there have been 6 Ebola viruses discovered, each an individual species within the Ebolavirus genus, and 4 of these can cause a hemorrhagic fever in humans. The first Ebola virus to be discovered was the Zaire Ebola virus, which is that which Mabalo had. In terms of symptoms, patients tend to at first suffer from a flu-like stage characterised by fatigue, fever, weakness, decreased appetite, muscular pain, joint pain, headache and a sore throat. This then tends to develop into nausea, vomiting, diarrhoea, abdominal pain and sometimes also hiccups. In many cases, though it has historically varied by outbreak, there is also internal and external bleeding. This can cause vomiting blood, coughing up of blood, and blood in the stool or even bleeding in the whites of the eyes. The mortality rate varies by outbreak and the level of care available, and has historically been anywhere between 25 to 85%, but hovers around 50% across all outbreaks and strains. The main cause of death is usually shock from fluid loss, and occurs between 7 and 14 days after first symptoms in most cases. The disease is spread between people by the exposure to the fluids or blood of infected individuals, most commonly through blood, feces or vomit. However, it has also been detected in a range of other fluids provided by survivors. For example, it has been shown to persist in semen samples for more than 3 months, and sometimes even as long as a year. It can also survive for a period of several days on a lone surface. Because of all this, although the actual method of transmission is quite controllable, at least compared to airborne diseases like the flu, it is still considered an extremely transmissible disease, and the CDC treats Ebola virus as a biosafety level 4 pathogen, the highest level. This means you need to run a level 4 rated laboratory to work on it, which are extremely expensive to build and run, and make research difficult. During outbreaks, part of what makes Ebola so difficult to combat is that it's found within a region where public healthcare standards are typically quite low, and yet effective treatment requires some of the most advanced precautions. Healthcare workers are often the first to become sick in large numbers, and the disposal of bodies in general presents another challenge, particularly in locations where traditional burial methods unknowingly expose fresh victims to infected bodies. In all, it's an extremely nasty and lethal disease that's very difficult to control in a public healthcare setting. Outbreaks tend to spiral out of control very quickly and can destroy whole communities. Across this video, we'll reference a range of outbreaks and interesting cases that give us clues about the virus's biology. We won't have time to pay the victims the proper respect in each case, but I just want you to hold in your head the brutal reality of this disease and what it means for the victims and their families. The events that open this video are those that began the 1976 Ebola virus outbreak in Zaire, which lasted for 26 days and was the instance in which Ebola was first identified. Interestingly though, it isn't the first Ebola virus outbreak that we know of. At the same time the disease was ripping through Yambuku, a similar disaster had begun around a cotton factory in Nazara, Sudan. This would eventually be identified as a genuinely separate outbreak of the same disease that had begun in June of that year. These two outbreaks provided to essentially be the warning shots of what Ebola would eventually become. Zaire's outbreak involved 318 cases and 280 deaths, while Sudan's had 284 cases and 151 deaths. From these outbreaks on, Ebola became known to healthcare workers as a transient killer, a rare but constant haunt in Western and Central African countries. From 1976 to 2014, there were an additional 13 outbreaks in the region. Each time the disease would appear suddenly and sporadically kill anywhere from dozens to hundreds. Although healthcare providers and researchers naturally took the threat seriously, it should be noted that compared to other tropical diseases or persistent healthcare challenges in the region, Ebola was just not as big of a killer. What did make it threatening, however, was its tendency to spread through healthcare facilities. In the countries where Ebola was commonly found, healthcare professionals were often forced to work with less than ideal equipment, which amplified initial cases within the community. For example, in the Zaire outbreak, one of the causes of the spread through the hospital was the reuse of needles between patients, most commonly when giving vitamin injections. This was a common practice not because healthcare workers were unaware of the risk, but due to the lack of resources, were forced to make a complicated decision in the name of public health. Other challenges, like a lack of PPE available for healthcare workers, meant that doctors, nurses and hospital attendants were often the first victims, removing the community's ability to provide effective care even at early stages of an outbreak. The worry was that in a severe enough outbreak, local healthcare services would be totally overwhelmed or even break down entirely, as had happened in the original Zaire outbreak. Then, in 2014, these fears came to pass.
This morning, I am declaring the current outbreak of Ebola virus disease a public health emergency of international concern.
An individual traveling from Liberia has been diagnosed with Ebola in the United States.
61 people die in this town. Everybody is affected now.
Nearly 1 in 10 people were killed in the span of a few months. Across West Africa, almost 1,000 people have now died in what has become the worst Ebola outbreak in history.
Sierra Leone has declared a 2-year jail sentence as punishment for harboring infected Ebola victims.
Now here's the hard truth. In West Africa, Ebola is now an epidemic of the likes that we have not seen before. It's spiraling out of control. It is getting worse. It's spreading faster and exponentially. So this is an epidemic that is not just a threat to regional security, it's a potential threat to global security. If these countries break down, if their economies break down, if people panic, that has profound effects on all of us, even if we are not directly contracting the disease.
The 2013 Ebola virus epidemic resulted in 28,646 official cases, causing 11,323 deaths across 10 countries, though the real numbers are likely much higher. The legacy of this outbreak is complicated and I think often misunderstood, but it's what propelled Ebola onto the global stage as a household name. Coverage of the epidemic dominated news cycles worldwide for months. Individual cases, particularly as they appeared in new countries, were tracked on an almost blow-by-blow basis. In a pre-COVID world, only Ebola had ever achieved this level of international scrutiny. Western audiences, often misled or encouraged by somewhat hysterical media, fretted over the threat posed by international transmission. In reality, the threat posed by isolated cases in wealthier nations with healthcare services equipped and able to respond was relatively small. Ebola, whilst very infectious, is spread mainly through the exchange of physical bodily fluids, making containment relatively doable in a well-equipped clinical setting. The true devastation was in the countries the epidemic had started in. Of the 11,000 deaths that I mentioned, virtually all of them were within Guinea, the country the epidemic started in, and the neighbouring nations of Sierra Leone and Liberia. All three of these nations were essentially overwhelmed by the virus. Although international aid did eventually flood into the region in the face of the developing humanitarian disaster, there's been a lot of discussion since around how slow the initial reaction was. For instance, despite the outbreak beginning in March, it wasn't declared an international emergency by the World Health Organization until August, by which time aid organisations like Doctors Without Borders were warning the situation was already badly out of control. If you want to do a little bit of interesting reading that criticises the initial response to the epidemic, this brilliantly titled article is a great starting point. It's difficult to describe the devastation within the region quickly and with the intention it deserves, and it's not the specific topic of this video, so we won't cover it in detail here, but in broad strokes, it was characterised by an almost total collapse in healthcare in the most impacted areas. The second major impact was economic, totaling 15% of the combined GDP of the three impacted nations, a significant burden to recovery. The epidemic lasted around 3 years in total, finally declared no longer emergency in March of 2016. Since 2014, there's been around 8 further outbreaks. Most are small with a death count in the dozens, with the exception of the 2018 outbreak in the Democratic Republic of Congo and Uganda, which killed just over 2,000. Efforts to combat this outbreak were heavily hampered by the civil war in the region at the time, and this outbreak didn't officially end until June of 2020, by which time the world's focus had shifted elsewhere. By June of 2020, Ebola had killed in all around 15,266 people globally since 1976, across 10 countries. It remains one of the most infamous tropical diseases following the 2014 epidemic, and is famed for its severe lethality and threat level. Following 2014, a huge amount of research has been poured into combating Ebola. Today, there is a greatly improved understanding of both how we can prevent its spread within the countries it is likely to be found, and treat it in a clinical setting. There are 2 specific monoclonal antibody treatments that have been shown to have positive clinical outcomes, and there are also 2 licensed vaccines, a global stockpile of which is maintained for future outbreaks. The academic review of Ebola ranges on everything from its attack vectors, that's how it actually enters our cells, the effect in the body, and its mechanisms of transmission. However, since 1976, there's been one prevailing mystery about Ebola that has simply never been solved. So, viruses are interesting. One of the longest running debates in biology is whether or not we can credibly consider them a form of life at all. Although they display some of the qualities we expect living organisms to possess, such as reproduction, evolution, and a genetic base, they also don't do a lot of things we would normally expect to see. For example, they have no cellular structure and lack their own metabolism, making them totally reliant on their hosts to survive. If you're unfamiliar, in a basic sense, all a virus is on its own is a length of genetic material, DNA or more usually RNA, covered in a protein jacket and occasionally also a lipid jacket. This full structure is called a viron, and it represents how the virus exists on its own. When infecting a host cell, the virus will have some method of invading the target cell and injecting its genetic material into the host. From there, the virus hijacks the host cellular machinery to achieve its own reproduction, usually creating many more virins in the process that go on to infect other cells. There's a lot of ways you can look at them. For example, some consider the affected host cell to be the actual living form of a virus, and the viron to be more like a spore stage of its life cycle, but it's often a matter of perspective. In any case, the key takeaway from this is that it means viruses that cause diseases in humans have to exist within a host somewhere for survival. If it ever ran out of hosts, the viron alone would be incapable of survival, and the viral population would eventually disappear. To an extent, this is true of all pathogens, that is, any natural agent that causes disease, be it a virus, bacteria, fungus, or prion, but is particularly true for viruses given their host dependence. This is important because understanding where a disease comes from gives us options in both treatment and preventing epidemics. For example, if a disease originates in a commercial livestock, at the start of an outbreak, a government could limit the spread by ordering a cull of livestock populations. Or, with diseases such as malaria, by understanding that mosquitoes are a key source of the disease, public health officials can take measures such as draining mosquito-infected swamps or issuing mosquito nets as a cheap and effective way to limit the persistent threat. All of this is a particular advantage for diseases where there is no effective vaccine or treatment, or the clinical instances of the disease are difficult to treat or particularly deadly. The natural source of a disease is called the natural reservoir. This term refers to the stable population of the disease-causing pathogen. Reservoirs can be populations of living organisms themselves, for example human populations for smallpox, or sometimes non-living environments, such as an infected water source for Legionnaire's disease. Some diseases may have multiple reservoirs, or a natural reservoir and a persistent infectious reservoir in human populations large enough to sustain it. In all cases, a key mechanism in the development of a disease outbreak is the jump a pathogen makes from its reservoir population to humans. This is done through a chain of infection that might itself have multiple steps and intermediate hosts. For example, a hypothetical disease where the reservoir is in a plant population, which passes to flies by feeding, which passes the disease to livestock by biting, which pass the disease to humans by consumption. These individual events are what cause new outbreaks of the disease to appear suddenly. This is particularly true for diseases which are extremely deadly in humans. Where a disease is deadly but not overly transmissible, it is unlikely it will survive in a stable populations in humans for long, as it will kill its hosts too quickly to spread far. This means it has to exist somewhere in nature, likely in a reservoir where it is either not deadly to the host or otherwise much less deadly, and it's only when a human comes into contact with the natural reservoir through a chain of infection that an outbreak begins. In a viral context, this usually means the host population species, which is functionally immune to the virus. So, let's bring this back to Ebola. What do we know? Since the original outbreaks in 1976, there's been ongoing research to understand the source of Ebola in humans and its natural reservoir, which has only accelerated since the 2014 epidemic. The evidence we have comes from a mixture of general patterns in the outbreaks to date, the lives and activities of individuals identified as the original case in each specific outbreak, such as Ebola, and organised field research and laboratory studies. At first, there are a few main clues to go off. First, Ebola is heavily localised to the regions of Western and Central Africa, giving a clear region in which to search for the reservoir. Second, outbreaks are extremely sporadic, often passing years between occurrences, suggesting that exposure events between humans and the natural reservoir were relatively rare. Third, the majority of outbreaks began with individuals who had travelled in rural areas, or were from small villages. Finally, there was also evidence from other similar diseases. I've already mentioned that Ebola was first thought to be an instance of Marburg virus, a similar hemorrhagic fever, which researchers knew could be spread by African green monkeys. If a similar disease was present in primate populations, that would be a good place to start. In the specific case of Marburg, there was also some extra evidence in the form of an additional case in 1975, where a tourist had contracted the disease while trekking in Zimbabwe. This tourist's movements in the time leading up to their infection had been carefully studied, and a range of possible exposure sources identified, including cave exploring, bushmeat consumption, and a strange bite they had received from possibly a spider. Now, when searching for the disease reservoir, you can essentially split the effort down into field studies, that's actually going out and capturing animals or collecting environmental samples, and non-field based studies, such as studying the disease in a lab or conducting a literature review of current research to clarify thinking across the field. With diseases, you also have the advantage of additional evidence brought by new outbreaks. If I bring you back to the timeline of Ebola outbreaks, one thing to note is that after those initial outbreaks in 1976, Ebola basically disappeared for 14 years before reappearing in 1999. Until this point, those initial clues were really all anyone had to go on. As part of the initial response to the 1976 outbreaks, there had been 2 studies aimed at identifying a reservoir, but it wasn't until 1979 that a research team began the first coordinated study of wildlife. As the resources weren't available to study Ebola alone, they paired it with a similar study aimed at exploring the source of human monkeypox, another disease found in the same region. The team collected animals from 2 sites in Zaire and Cameroon through a mixture of trapping and purchasing or bountying animals from local villagers and hunters. In Cameroon, the research team acquired virtually all of their animals from semi-nomadic hunters who travelled between villages supplying bushmeat to the local region. In total, they collected 1,664 animals, mostly from Zaire, representing 125 species across primates, bats, squirrels, birds, reptiles, and other rodents or mammals. The aim had been to try and collect 50 of each species to cover off the chance that Ebola exists only in very low concentrations in an animal population, but this had only been achieved for 16 species. Unfortunately, the study drew a blank on identifying any species that had Ebola-specific antigens, something that might identify them as being carriers. The researchers noted in their paper the weaknesses with both the technique they had to rely on in these tests, but also the challenge of collecting animals in such a non-random manner. The majority of the animals they had collected were those most easily caught, the one humans came into contact with a lot. Given the rarity of Ebola outbreaks, the author felt it was unlikely for one of these species to be the culprit. In 1994, Ebola resurfaced. Between 1994 and 1996, there were 3 back-to-back outbreaks, 2 in Gabon and 1 in Zaire. 2 outbreaks fit a previous pattern, with the first patients being rural workers, 1 from a gold mining camp in Gabon and 1 a rural forest worker from Zaire. The third, however, started when a group of children in Gabon found and butchered a chimpanzee carcass before transporting it back to their village. This carcass was later found to have been infected with Ebola. Now, hearing this, you might be tempted to think that monkeys are a pretty good culprit for the Ebola reservoir. After all, the closely related Marburg virus was associated with infected green African monkeys, and subsequent Ebola outbreaks after 1996 were also associated with the consumption or exposure to primate carcasses, particularly chimpanzees. However, scientists discredit this idea for one fairly simple reason. Ebola virus is extremely lethal in primates as well as humans. In fact, alongside an outbreak, it's common to see an associated die-off in primate populations in the same region. This disease is essentially too lethal in these populations for them to be the source. This is the same reason those monkeypox researchers in the 1979 field study were looking for a reservoir as well. The disease is too deadly in monkey populations to survive long term. In fact, in 2003, zoologists began to ring the alarm that ape populations in western equatorial Africa were suffering a catastrophic decline, having halved from 1983 to 2000. Most of this was caused by hunting and habitat destruction, but the third most common cause was Ebolavirus spreading through gorilla and chimpanzee populations. Interestingly, this strain of the virus was also specific to non-human primates. The 3 outbreaks between 1994 and 1996 did, however, provide one more clue. The disease's genetic code had remained very stable, as in it was very similar to the 1976 outbreaks. This suggested that whatever the reservoir was, it was very likely a small population, or at least the disease only existed in a small number of the host animal. Otherwise, researchers would have expected to see a much greater degree of mutation over time. In 1995, there were 2 field studies in Zaire done following the latest outbreaks. One focused on vertebrates, that's any animal with a spinal cord, and the other on arthropods, insects, spiders and such. The studies followed a similar methodology to the previous, trapping small creatures and purchasing larger ones from locals. Across 3,066 specimens in the vertebrate study, and 34,985 in the arthropod study, once again, they drew a blank. Another study in 1995, this time in the Thai forest on the Ivory Coast, where Ebolavirus had been detected by researchers investigating a collapse in the chimpanzee population, also found nothing. Apart from these field studies, at the same time a large amount of laboratory study was poured into identifying Ebola's natural reservoir. A series of studies on Ebola and other similar filoviruses involved attempting to infect a variety of species with Ebola and observing their response. These studies found that some suspected plants and arthropods to be a blank, but that some species of bat were capable of sustaining the infection, and that some small animals like rodents showed that they had possibly encountered filoviruses at some point in their evolutionary history. Bats in particular are an interesting culprit, because due to a variety of reasons they seem to make particularly good hosts for human diseases. They're mammals, so they're similar enough to us that their disease can impact us too, but different enough that those same diseases often don't have the same impact in bat populations as they do in humans. In fact, they're a target of suspicion so often in new diseases that their presence in both Zaire bushmeat and the roof of the Sudanese cotton factory that was the site of the 1976 outbreak was noted. Apparently there was a colony living in the rafters. Really interestingly, a study from 1996 also showed that some fruit bats were capable of carrying the disease asymptomatically for up to 4 weeks at a time. Then, in 1999, there was finally some field evidence. A new study performed this time in the Central African Republic used a new technique on top of the antigen analysis used in previous expeditions. This time, reverse transcription PCR was used on RNA and DNA extracts from the organs of 242 small mammals. In 8 small rodents, they found sequences associated with Ebolavirus. This finding didn't prove that these rodents were in fact the reservoir, but it did suggest they had been in contact with the disease at some point, and clearly survived well enough afterwards. However, probably the main takeaway was the researchers showed how difficult it was to isolate sufficient samples from their specimens to provide accurate testing. PCR in this instance was one way to potentially screen for the disease more effectively. The 2000s brought yet more outbreaks of Ebola, albeit small ones, this time focused once again in Gabon and the Republic of Congo. From 2001 to 2003, a new field study was once again conducted. This time making use of the new PCR method, the researchers were able to find the strongest evidence to date of a reservoir species. Across over 1,000 vertebrates, 3 species of fruit bat were identified as having Ebola-virus-specific antibodies using the normal method, and by PCR, also showed Ebola-associated sequences in liver and spleen samples, 2 organs that are heavily impacted by Ebola virus. The authors then returned to the region with a renewed focus on the bats as a candidate, and conducted a large-scale survey of the 3 potential reservoir species they'd identified. They captured an additional 1,390 bats across both Gabon and the Republic of Congo. This time, 40 bats were shown to be positive for Ebola virus antibodies, distributed across all of the sites, including both those with active Ebola virus outbreaks and those without. The study authors also drew some interesting speculations from the data, noting the prevalence of these antibodies rose and fell over time by region, and suggested that by studying these fluctuations, we could see when spillovers into human populations were likely to happen. However, it's important to note that none of this is direct proof these bat species are the reservoir. The research very heavily implicates them as being capable of being asymptomatic or low lethality hosts, but actual proof would require you to isolate a living sample of the disease from an animal. For the Marburg virus, this was actually achieved in 2009. There had previously been a very similar study to the Ebola bat survey for Marburg in 2007, which had found similar results. A later study in 2009 that focused on an Egyptian fruit bat population in Kitaka Cave, Uganda, the site of a Marburg outbreak, actually succeeded in isolating live virus from bat tissue, and an active infection rate of about 5% in the resident bat population. Really terrifyingly, this bat population is actually pretty widespread as it's migratory, and goes far beyond what was originally thought to be the range we would normally find Marburg in. This has been a focus of quite a bit of more recent research on Ebola and other filoviruses, where tested fruit bat populations have found Ebola virus-resistant bats in regions we did not know it to be present, such as Bangladesh. Since 2007, research on the Ebola virus reservoir has continued pretty much full force, particularly in the wake of the 2014 epidemic, but there hasn't been a huge amount of movement since. Following the 2014 outbreak, another species of bat, the Angolan free-tailed bat, an insectivore instead of a fruit bat, was added to the potential reservoir list. This was after a study conducted on the origins of the 2014 epidemic, which began with a 2-year-old boy named Emile Uwamono in the village of Meliando, Guinea. This was unusual, not just because it was the first emergence of Zaire Ebola virus in Western Africa, but because there was no known large colony of fruit bats nearby, or were large animals that were suffering an Ebola virus-related die-off. Primates also weren't a common local bushmeat due to low populations in Guinea, and most of the bushmeat was actually sourced from quite far away and smoked. Bat hunting, however, was relatively popular, particularly amongst children of the village. The researchers conducted a study and captured several fruit bats in the village, showing that migratory species were at least present and close to human populations. However, the research team still suspected non-fruit bats to be the main culprit, as the opportunistic hunting of fruit bats in the migratory season was still pretty rare. The team performed a survey of the surroundings of the village, and just 50 metres away from Emil's house, found a hollowed tree that had been burned. This tree was next to a path women would use on their way to the stream to collect water, and villagers described how children would frequently play in the tree and even capture bats. When it had caught fire, they had described how, quote, a rain of bats had fled from the tree. Testing the ground around the tree, they were able to recover enough genetic material to determine the colony had been that of the Angolan free bat. So, where does this leave us? Today, opinion amongst the scientific community is largely divided into one of two camps. The first belief is the bat theory, the idea that there's a persistent, low-level infection in one or multiple bat species in Central and Western Africa. Outbreaks in humans begin when the bats are captured and eaten, or somehow pass their disease on to monkey or deer populations which are then eaten in bushmeat, possibly by feeding on the same fruit. The second is that there is an as-yet unfound reservoir out there that is actually the main source of the bat infections, who are only incidental hosts, for example a tick or other small arthropod that somehow infects the bats themselves. It's worth noting that this is the opinion of jean-jacque, a virologist whose book on filoviruses is essentially the single best record of study in the field. The logic that underpins this view is that the rarity of outbreaks versus the commonality of bats in bushmeat and human settlements suggests that bat populations are only occasionally infected with the virus. This might explain why live Ebolavirus has never been identified in bats. The same could also be said for the low incidence of antibodies and viral DNA, suggesting only periodic infections at the population level. Whatever the case, research in this field continues. Field studies are extremely difficult to run, and even more difficult to fund given their expense. You have to take a lot of equipment into rural areas, not helped by the fact that the modern territories home to Ebolavirus are frequently unstable, such as the Democratic Republic of Congo, which is home to one of the world's longest-running civil wars. In the aftermath of an outbreak, field studies can also be controversial. The victims of a savage outbreak might be frustrated to see the international community taking a greater interest in bat populations than the well-being of a ravaged community. The low chance of success also makes the required expense difficult to stomach for most organisations, particularly when the suspected level of infection is quite low. Research in labs and academic papers has continued. For example, a paper from early this year showed that free-tailed bats were capable of vertical transmission of the virus, meaning that a pregnant bat would pass its disease off to its offspring. This is important because it's something we'd normally expect to see in a reservoir species. Further, zoologists have been adding to the ecological evidence by comparing the range of bat and small mammal populations to more recent outbreaks to show a fairly strong correlation. However, the critical smoking gun continues to elude the wider scientific community. Until it's found, we simply don't understand the actual source of Ebolavirus, or fully how to prevent future outbreaks. For now, it's still out there, and the secret is nestled deep, somewhere in the countryside of Africa.